Hello and welcome. I want to spend a few minutes talking about one of the most important proteins that we find embedded in cell membrane, and that is the sodium-potassium pump. This protein is found in almost all animal cells and is critical for some of the core functions of cells. For example, it is responsible for giving cells their resting potential. This means that it gives cells a resting electrical charge that is very important for example, in neurons. This resting potential is critical to allow the formation of action potentials, and those action potentials are how we move information around in neurons in the brain. Secondly, the sodium-potassium pump plays a critical role in controlling cell volume. Because this pump moves ions inside the cell outward and outward inward, it can control the internal osmolarity of a cell, and therefore, by osmosis, the volume of fluid that follows this ionic gradient will determine the size of cells, or their volume more specifically. And finally, and interestingly, or in fact not finally, there's some other smaller functions that we're not going to talk about, but the third major function is the sodium-potassium pump also provides mechanisms by which the transport of non-ionic components can be moved across cell membrane in a sort of co-transported mechanism. So these are the key functions of the sodium-potassium pump. What does this pump look like and how does it work? Well, let's remind ourselves that we have cell membrane, and that cell membrane is made of phospholipids, and I'm not going to draw them all. I'm running out of steam drawing all these phospholipids each time for cell membrane, but remember it's a bilaminar structure that forms the cell membrane. The sodium-potassium pump is a protein embedded in this cell membrane. Now, the word pump is often used because what we're going to do is we're actually going to move ions against the concentration gradient. So these ions in the normal state, you see a high concentration of sodium on the outside and a relatively low concentration of sodium on the inside, and you see a low concentration of potassium on the outside of a cell and a high concentration on the inside. So a pump is going to allow us to move ions against the natural gradient. If you think about the sodium, the natural gradient would see diffusion happen from the outside inward, and it would naturally have a tendency for potassium to move from the inside outward. But a pump, because this pump uses energy, and that is through the conversion of ATP to ADP, because there is energy consumed in this process, the ions are moved against the gradient. And in fact, in this situation, what we find is 3 sodium ions on the inside attach to the pump through the addition of energy. These 3 sodium ions are moved in conformational change of the protein. They are moved to the surface, the external surface, and then expelled into the external environment. Once they're expelled into the external environment, 2 potassium ions attach, and those potassium ions then, through conformational change in the molecule, are then moved to the inside of the cell and expelled. So what we end up with is, for every 3 sodium that are sent to the external environment, 2 potassium are sent back to the internal environment through the use of energy provided through the ATP-ADP. This moves the ions against their gradient. Do you notice we're taking from the low gradient concentration side of the membrane and adding to the high, which is a critical part in this process. And the other important thing to notice is, by moving 3 relative to every 2, we end up with an ionic misbalance, remembering that sodium is single positive, as is potassium. I'm just going to turn those to yellow so we keep the track of the conversation. There we are. So by moving 3 outward versus 2 inward, we end up with a net ionic balance that will make the external environment positive and the internal environment negative. And this is what we call the resting potential of the membrane. The resting potential sees the external environment positive and the internal environment negative. If you remember back to what I said in my list of functions, we have now covered off the first function here of resting potential. The resting potential is a natural event that follows the 3 out, 2 in rule that happens with the sodium-potassium pump. The next one we'll look at is cell volume. What is happening with cell volume? Well, clearly, if you you regulate the rate at which ions are moved in and out, it's the total number of ions, so it's the sum of all sorts of ions and proteins. It's the total sum that is the key to determining the ionic balance, determining the osmolaric balance. So the total sum of these particles interiorly will determine the movement of water into or out of the cell. Now, I've drawn this as a wiggly line because actually the movement of water across a membrane is not directly through a membrane, it's actually through some other proteins, but we'll talk about those in a separate discussion. So cell volume is controlled to some extent by the action of the sodium-potassium pump through its ability to change the total quantum of particles, ions, proteins, etc. inside the cell, and the water will then follow that or go the other direction depending on this concentration. And I'm going to leave for a separate short video a discussion of the transport potential that is produced by the sodium-potassium pump. And just as a final interesting fact, the sodium-potassium pump in all cells of the body use up about 20 to 25% of the total energy of a cell is used in just this part of continuously keeping the sodium-potassium pumps active. And in fact, in neurons, this goes above 50%, and it's argued that above 60% of total energy capacity is used purely to keep the sodium-potassium pumps active and to keep the gradients as they are.